The Molecular Bases of Inheritance, presented by Lucy Late. In this assignment, I will be talking about the structure of DNA, the process of DNA replication, the structure of chromosomes, and the karyotypes in animals. The structure of DNA. DNA is made up of molecules called nucleotides. Nucleotides are the basic format of DNA, comprised of one group of phosphate, one group of sugar, and one base, and a base of nitrogen. There are 4 different types of nitrogen bases: adenine, guanine, cytosine, and thymine. Out of each of these 4 bases, only two join together at a time, and the ones that join together are specified. This means that only A and T connect and only G and C connect to one another. This is called base pairing. If one of these nucleotides is damaged, it causes mutations. This is what the structure of DNA with the nucleotides looks like. It is similar to the structure of a ladder. DNA replication. This is the basis of biological inheritance. This is where DNA makes a copy of itself during cell division. However, there are a few steps to DNA replication. There is a series of steps involved in the process of DNA replication. Firstly, the double helix structure of the DNA strand must unzip or unravel, and to do this, an enzyme called the helicase is involved. This enzyme breaks down the hydrogen bonds holding the bases of DNA together, A plus T and C plus G. This is pictured here on this image as the blue triangle separating the bonds. Further on, you can see purple spherical structures on the sides of the bonds. These are called single-strand binding proteins, and these block the DNA from joining together again. DNA polymerase then fill in and create a whole new strand of DNA by adding new nucleotides. After the polymerase create the new strands of DNA, the DNA ligase then seals the bands back together, and as this happens, the strand automatically winds back up again. The end result is 2 new DNA chains, one containing new nucleotides and one containing old nucleotides, meaning that half of each chain contains new nucleotides and half contains old nucleotides. Chromosomal structure. What is a chromosome? A chromosome is a thread-like structure made up of nucleic acids and proteins. The chromosome is found in the nucleus of most living cells, as this is where genetic information is stored, such as genes and DNA. The chromosome is made up of 2 identical chromatids joining together, both carrying DNA by the histones. Histones compact the DNA into the chromosomes, as pictured here. The centromere is the point where both of the chromatids are bunched together, creating this X shape. Here is a more in-depth image of what a chromosome is made up of. DNA and histone proteins are packed into the chromosomes. The centromere, or the point at where the 2 chromatids join together, give the chromatids its identifiable shape, whereas the P arm, the shorter arm, and the Q arm, the longer arm, both give a clue as to where specific genes may be. Karyotypes. What is a karyotype? A karyotype is the complete amount of chromosomes an animal has naturally within its body. The amount of karyotypes differ from animal to animal, e.g. a horse and a cat. Whereas the horse has 32 chromosome pairs in its body, the cat has only 19 pairs. It is important that animals have a set amount of chromosomes in their body, as without the set amount, low vitality and infertility can arise. Here is a more in-depth image of cat karyotypes. The cat has a total of 19 pairs of chromosomes in its body. This makes up the number of karyotypes found naturally in a cat. And here is a more in-depth image of the karyotypes that are found in a horse. Whereas cats only have 19, Horses have 32 pairs of chromosomes in its body. This makes up the horse's karyotypes.